Hi friends and welcome to my channel Be Yourself. This is Dr. Rajni Sharma and today I'm going to deal with some other questions of part B CSIR net examinations, especially of life sciences. In my last video I have discussed few of them. Let's start with some other questions in this section. Here the first question is that choose the most appropriate pH at which the net charge is 0 for the molecule from the data shown below. Here we have to find out the pH of that structure whose net charge is 0. If we will see this structure, then it has net positive charge. Come to this structure, it has one negative and one positive. It means the net charge will be 0. Then again move to the next structure. Here it will be one more negative charge. It is not visualized clearly, but it is negative charge. Okay, so in this structure it has net negative charge, and this too it has negative charge. So we have to find out the pH where this structure will exist. To calculate this pH, what we will do, we will take the average of these first two pK. Let's calculate 2.02 plus 3.80 by 2. When you will solve this, you will get 2.91. Now the question is that why we are taking only the first two pK value, not the third one. This will be more clear if you will see this graph of the lysine. Here too, if you will see the pKi, the isoelectric point is lying in between this pK2 and pK3. It means in this structure which has the net charge 0, because one negative charge and one positive charge, net is 0. So what we do, we can take the average of its nearby pK values to calculate the isoelectric point of any molecule. This also applied to other amino acids. Hope you have understood this, how this come. Now move to the next question. Choose the correct statement about the peptides in Ramachandran plot. First is the peptides that are unsaturated will have all the backbone dihedral angles in the disallowed region. In the first option, you have come across two terms. One is the dihedral angles and other is the disallowed regions. So let's first discuss over this, then you yourself will be able to answer these questions. I will not answer this question. You yourself will able to answer this question. So let's start. This is the linear structure of the peptide chain. I have shown it two-dimensionally because it is very difficult to it was difficult for me to represent in the three-dimensional way. Here you will see there are the three unit of amino acids. There will be one R3 will be here. This is the side chains, and the carbon which is attached to the carboxyl, and one side with the ammonia is the C alpha carbon. So there are three amino acids which are connected with each other with the help of peptide bonds. Okay, there are two peptide bonds in this structure. Here is the C terminal ended with the carboxylic group, and here is the N terminal and with the amine group. So what I want to tell you that if you will see, I'm dealing with only this peptide group. You will see here, we know that oxygen has lone pair of electron. This being more electronegative than carbon, it will attract electron towards itself. Also, nitrogen also has a lone pair of electron. To compensate the electron which was extracted by the oxygen from the carbon, this lone pair will be donated to the carbon or the NC bond. Because of this reason, a partial double bond gets developed in between C of the carboxyl carbon and this amine nitrogen. So this the peptide bond will be remain as it is, but these two bond between and C alpha C, it is free to rotate. Okay, now this is not a in a linear way, rather it will be in the tetrahedral form. And when you will see, if it is in three dimensional, you can't draw a line for this polypeptide chain, rather there will be a plane. It we can represent in a definite plane. And the rotation between C alpha and N is represented by phi, and C alpha C represented by psi. And when we will plot a diagram for the angles of psi and phi, this plot is known as Ramachandran plot. Actually, this shaded region, we don't get this particular shaded region, we actually get the dots in this regions, and there we will calculate the where the most dot will lie. From the dots we can analyze that the particular uh, peptide is what the structure of the particular peptide. For example, most of the if any in a peptide, most of the dots lie in between this region, then we can say that this peptide will have the anti-parallel beta sheets structure. Similarly, when the more dots will accumulate in this region, then we can say that the polypeptide has a parallel beta sheets. Likewise, there are some other structures too, or the areas where the angles of psi and phi can lie. Actually, the psi and phi angle lies in between plus 80 to minus 80 degree. Okay, we can able to predict whether the polypeptide is alpha helical, beta sheets, or we can even analyze a particular type of beta sheet structure from this Ramachandran plot. Now, second word was disallowed region. What is that? In this figure, this is also a Ramachandran plot. The white region shows the disallowed region, while the red and this yellow shaded region is the allowed region. What does it mean? I'm taking here the example of water. We know that water has a tetrahedral structure, one oxygen and two hydrogen atom, and oxygen has two lone pair of electron. Okay, I haven't drawn this figure so nicely, so excuse me for this. If it is in tetrahedral form, then the angle should be in between hydrogen, oxygen and hydrogen should be 108 degree, but it is not so. It is only 105 degree. This is because of the repulsion from the lone pair. It has decreased the angle because of the lone pair repulsion. Likewise, in peptide chain also, the R group, which is also in a side chain, which determine the particular nature of the particular amino acid, whether it can be polar, non-polar, acidic or basic in nature, determine the phi and psi angle, or the rotation between C alpha and C alpha C, which is here it is phi and it is psi. Okay. Uh, the first option was in all the unstructured peptides. It doesn't mean that unstructured peptides may have some amino acid whose angles can lie in the allowed region. It is not possible that it will completely all the angles will be in the disallowed region, but the condition will be that the most of the dots will be in the disallowed region. The only exception is that the glycine mostly like in the disallowed region. This is because the R group of the of glycine has H. So there is no steric hindrance because of this H group in comparison to the proline, which has very bulky group and other amino acids too. So now back to the question. Again, you come to the options. The peptides that are unstructured will have all the backbone dihedral angles. And what was dihedral angles? Sorry, I forgot to tell you. The phi and psi angle is known as dihedral angles. Okay. In the disallowed region, you come to know about the disallowed region. The yellow region and red region was allowed region. Yeah, in this red region, there will be the asterisk hindrance will be nil, but in yellow region there will be somewhat asterisk hindrance, but that can be overcome, or that can be in the that can form a particular structure there, or we can say that are allowed. That much of asterisk hindrance is allowed in a structure. Okay, now so this is not possible. All the unstructured have the dihedral angles will be in the disallowed region. There may be possibility that they can be in the allowed region. Okay. Second point is that it is not possible to conclude whether a peptide adopts entirely helix or entirely beta sheet conformation. There was clearly mentioned that the reason, which reasons can be in the beta sheets or alpha helical region or the beta sheets. So this is also not true for the Ramachandran plot. Third is the occurrence of beta turn conformation in a peptide can be deduced. This is true because in the figure I have shown you that how in different regions the beta sheet is there, but even in the beta sheet region how different structure of the beta sheet can be deduced from the Ramachandran plot. The last is that sequence of peptide can be deduced. It is not so. We can deduce the structure of a peptide, not the sequence. Okay, so the answer will be 3. I think you will be satisfied. Now move to the next question. It's very simple. The question is that equilibrium constant KEQ of a reaction is the ratio of product to the substrate concentrations, and the relation between KEQ and the free energy change in the reaction represented by del G is as follows. This is the formula, and for the reaction A the Ke value is 10, and for the reaction B K equilibrium or the equilibrium constant is 100, and from this all this data we have to find out which option is correct. Before that we are analyzing here the del G or the Gibbs free energy of reaction A with that of the Gibbs free energy of reaction B. So let's first calculate the relation between A and B in respect to its Gibbs free energy. Here we have given equilibrium constant. We can also write it as log 10 of K equilibrium, while the rest will be the same, minus RT. It means the lesser the value, because here minus sign is there, lesser the value, more will be the del G. It means when we will calculate Ke for the equilibrium constant for reaction A, what will be the log of 10? It will be 1. So we can get only minus RT. And the equilibrium constant for reaction B, it will be 10. What will be the log? We can write 100 as log 10 to the power 2. So it will be, when we will solve is, we can write as 2 log 10. So this was for reaction A, and the reaction B it will be 2RT for B. As minus sign is here, so reaction B will be less than, or the Gibbs free energy for the reaction B will be less than the del G of reaction A. So let's see which option should be there. Option A is greater. Okay, so this will be del G of reaction A will be greater than del G of reaction B. Next question. Excess oxygen consumed after a vigorous exercise is, here the option first is to pump out lactic acid from muscles, to increase the concentration of lactic acid in muscle, to reduce dissolved carbon dioxide in blood, and to make ATP for gluconeogenesis. First have a look what does it means, consumption of excess oxygen. Actually it is the excess post exercise oxygen consumption, or it is also known as after burn. what happens when we perform exercise? Our body required or get deposited of the lactic acids. Okay, so when we come after exercise, when we come to rest, there is the increase in the demand of the oxygen, and this is also known as oxygen deficit. Okay, our body get deficit of oxygen because whatever the required, suppose when you are hungry and you have or when you are not hungry and add some of more and more foods, and now when you become hungry, really want to eat, and when you don't have the food for the consumption, then that condition is known as food deficit for you. Likewise, when our body uh, has consumed more and more oxygen while doing exercises, now when it really want oxygen for the metabolism of some material which is deposited during the exercise, then that condition is known as oxygen deficit for the body. Okay, during this process or the post exercise, what our body do, it inhale more, or it increases the intake of oxygen to balance the hormone in our body, to replacement of the fuel storage, cellular repair which has damaged during our exercise, and innervation. What is innervation? For the nerve supply to the desired body parts, and the anabolism means for the construction process. And apart from this, it also synthesizes some new ATP, and it donates the ATP to the phosphate group, and until the ATP and creatine levels are back to the resting state level again. Okay, what happens when we do exercise or the strenuous exercise, when our body started taking more and more oxygen, then on that time, in the case of our body or the cells started anaerobic respiration. And what happens during anaerobic respiration? Lactic acids form instead of pyruvate, and these lactic acids go on to the livers, kidney, and cardiac muscles where they get deposited. Okay, and this is not good for the health. So these cells now convert this lactic acid into pyruvic acid. I have discussed some point regarding post exercises requirement of the oxygen. Now I think you can yourself can answer the questions, which was, sorry, the question was the oxygen consumed after a vigorous exercise was that to make ATP for the gluconeogenesis, that is not to pump out lactic acid. It doesn't pump out lactic acid from the muscles, rather it changes the lactic acid into pyruvic acid in the muscles itself. Okay, and from there it can go to the Krebs cycle. Okay, so the answer will be to make ATP for the gluconeogenesis. Now move to the next question. It is very simple. Which of the following describes the primary function of flippases? Flippases has a very primary structure, that is when our body synthesizes phospholipids. Actually what happens, phospholipid is synthesized, or the lipid is synthesized in the smooth endoplasmic reticulum in the cytoplasmic side. Okay, this cytoplasmic side and this is the luminal side. Uh, when it gets synthesized in the cytoplasmic side, there will be the high concentration of the phospholipid toward this cytoplasmic side. Then what happened, along with the consumption of ATP, Flippy's enzyme distribute it to the inner side of or the luminal side of the endoplasmic reticulum, and from here it can transport it to other where it is required. So the answer is very simple, that is move certain phospholipids from one leaflet of the membrane to another, which is its primary function. Next question. Mitotic cyclin-CDK activity peaks in M phase. This is because mitotic cyclin is synthesized only in M phase. Threshold level of mitotic cyclin accumulates only in let G2. Cyclin subunit is activated. Let it be weak for you read yourself the options. I am discussing some more points regarding this cyclin-CDK activity, especially when it is entering into the mitotic stage. What happened? In the G2 phase, cyclin B is synthesized, and when it's synthesized, it will bound to the CDK1. And CDK1 has three sites: threonine 161, tyrosine 15, and tyrosine 14, which is phosphorylated. Okay, when threonine 161 is phosphorylated, it will activate cyclin B. But when tyrosine 15 and 14 is phosphorylated, it will deactivate the activity. And here the tyrosine 15 requires V1 factor for its phosphorylation. Then when actually this is a checkpoint, when our body gets checked with all the requirement sufficient for the cell division, then CDC, then there will act CDC25C, and it will dephosphorylate tyrosine 14 and 15 positions, and now it again get activated and it is able to pass to the M phase. What happened after dephosphorylation? It is passing to the M phase. Okay, and in the M phase, what happened? Again, cyclin B is degraded and it will pass to the its raw material can be again used for its resynthesis in the G2 phase. So this is how the cycles carry out, or the checkpoints is carry out. Okay, now come back to the question. Mitotic cyclin synthesized only in the M phase. It was not there. It was synthesizing in the G2 phase. So it will be not there. Threshold level of mitotic cyclin accumulates only in G2. It is not so. Cyclin subunit is activated by phosphorylation. It is not activated by the phosphorylation. Rather, it was activated kinase activity, which was CDK1, was activated by dephosphorylation in the M phase. So the option will be fourth one. Okay, next is also very simple question, that is not much explanation is needed in this. The gel to liquid crystalline phase transition temperature in phosphatidylcholine lipid composed of dioleoyl, dipalmitoyl, diasteroyl, and palmitoyl alcohol fatty acid is increasing order can be. Actually, each of the certain phospholipid has its own transition state from the gel to liquid, and when we will see this, the option 2 will be correct. Actually, it is the standard one. All has approximately DSPC have approximately 54 to 55 degrees Celsius, where it can change from gel to lipid crystalline, and DPPC can change has 40, sorry, it should be only 42 degree Celsius, and POPC has -2 degrees Celsius, and DOPC has 16.5 degrees Celsius. Okay, so the option will be 2. Apart from phospholipid itself, the cholesterol in the membrane are responsible for the transition from gel to liquid. It rather prevents, or it maintains a proper or we can the fluidity of the membrane. How? When the temperature get increased, it will hinder the movement of phospholipid, and it will prevent it from uh, uh, in, or we can say it separates, or it prevents it from decreasing the fluidity of the membrane, and when temperature gets decreased, it will prevent it from freezing. Okay, next question is that which of the following is not the example of transmembrane transport between different subcellular compartments? First is the transport from the cytoplasm to the lumen of endoplasmic reticulum, transport from endoplasmic reticulum to Golgi body, transport from stroma into thylakoid space, transport from mitochondrial intermembrane space into the mitochondrial matrix. The answer will be transport from endoplasmic reticulum to the Golgi complex. Why? What does the transmembrane transportation mean? Suppose this is the membrane, and transportation across the membrane is the transportation membrane. In all the previous cases, if you will see from cytoplasm to lumen or from the other cases, there is a definite membrane across which the solutions or the materials is passing across the membrane. But transportation from endoplasmic reticulum to the Golgi complex, what happens? Suppose it's endoplasmic reticulum, okay, and Golgi body. We know that after the post-translational modification, the required protein get transported to the Golgi body, and from Golgi body to the place where it is needed on the basis of their selective markers. Okay, what happened? Suppose this is the desired protein. It will first express on the endoplasmic reticulum. After that, a vesicles will be pinched out, and this vesicles will fuse with the endoplasmic reticulum, and now it will express in the endoplasmic reticulum. Okay, so it is not crossing the membrane, rather it is fusing with the membrane. The vesicle is having the desired protein is going to express on the Golgi body by fusing over it. So this is not the transmembrane, rather it is a vesicular transport. This was in this video. I have discussed only this much questions. I will discuss more questions which was left in part B in my upcoming videos. So don't forget to subscribe and share my channel, and if you found that somewhere I'm wrong in this explanation, then do comment me on my comment in my comment box. I'm waiting for you, because I'm also a human being and I am not very much expert in each and every field, so I can be wrong somewhere. So don't hesitate to comment me on the comment box, and if you want to give some suggestion or some more topic, then you can also write to me in the comment box. Till then, goodbye and all the best for your future examination, and be yourself.